to chapter 16. Chapter 16 is talking about proteins. But first, before we start talking about proteins, we are going to talk about amino acids. So just like the rest of our other major macromolecules, like our carbohydrates and our lipids, we have the single individual piece, but then you can put them together to make larger structures, such as your lipid bilayer when your lipids aggregate together, or your polysaccharides when you make those glycosidic linkages between individual carbohydrates. With proteins, we actually string together what are called amino acids. So that's the first thing that we're going to talk about when we look at this particular chapter. So we will overview looking at the 20 amino acids that we have, and then we will talk about peptides. So peptides are when we put those individual amino acids together. Then we will talk about proteins. So proteins are also known as polypeptides, so many different amino acids or many different peptides that are put together. The other thing to remember, if you don't remember anything from this chapter, please just remember that for proteins and for all major macromolecules, frankly, their structure dictates their function. Therefore, if you have the wrong structure, you have the wrong function. So we will talk about how we get to protein structure, what protein structure enables us to do. We'll give a few examples of different types of proteins. So we have structural proteins, and we have functional proteins. And then we will go into enzymes. So enzymes are a particular type of protein that actually catalyzes a chemical reaction in our bodies, for example. So we will take a look at how these are regulated, because it's very important. It's probably not a good idea to have all of your enzymes on at the same time, but you also don't want all of your enzymes off at the same time. So we talk about there are different metabolic pathways. Maybe you've heard of like glycolysis or the Krebs cycle or the TCA cycle. A lot of these have enzymes that sometimes they need to be on and sometimes you need to shut them down depending on what your body's communication signals are. So let's start with those amino acids though. So if we take a look at our amino acids, that's how we get protein. So when we covalently link, so remember, with carbohydrates, we make that glycosidic linkage to make disaccharides or polysaccharides. So that's an actual covalent linkage. With lipids, they actually aggregate together. They don't actually physically bond. But then we're back with amino acids. We string and covalently bond amino acids together to give us proteins. So again, the biggest thing though is structure dictates function with proteins, which means if you put in the incorrect amino acid, you might get different properties, different structures, and therefore different functions, or a completely dysfunctional protein. So a great example, like you can see and that your book talks about, that you can see in the slides here, is something called sickle cell anemia. So sickle cell anemia, we'll refer back to it again in chapter 17, talking about DNA, but if you have a mutation in your DNA, it can lead to a substitution of a certain amino acid with another amino acid. And when you do that, it can lead to the overall different structure of the protein. So as you can see in the slides, we have the quote-unquote normal hemoglobin in A and what our normal red blood cells look like. But if you have a sickle cell anemia, or if you have a sickle cell disease, for example, you have a mutation and you put in the wrong amino acid. And as a result of that, you get incorrect folding, incorrect structure of the protein, like you can see in part B there. That leads to these distended or sickled looking red blood cells, which ultimately leads to the disease. So here's the thing about amino acids that I like to say is biochemists are not creative people. So when we named an amino acid, we named it because it has both an amino group, if you remember from your organic chemistry terms, and a carboxylic acid group, okay? Or an amine and a carboxylate, which is a decarboxylated amino acid, okay? So again, if you're taking a look, you have over here, this is an amino group. And then over here, this is a carboxylate. Or if you had this also protonated, right, if you had COOH instead of COO minus, that could be carboxylic acid. So that's where we get the word amino acid when we're looking at the structures of these. So here's the nice thing about these 20 amino acids. Yes, you do need to know the differences between the 20 amino acids. However, the only thing that differs is this one group off of this center carbon here. So if you remember in carbohydrates, we talked about chirality. Well, this carbon right here, with one exception for 19 of the 20 amino acids, this is a chiral carbon, so 4 different groups. Okay, so every single carbon in the center here, sometimes you might see a little alpha. We call that the alpha carbon. So if you ever hear me refer to that as an alpha carbon, this is the carbon specifically that we mean. This carbon always has a nitrogen, always has a hydrogen, and always has a carboxylic acid group or carboxylate group. So that is always the same with all 20 amino acids. The only difference are these different R groups. So those are the things that are the difference between those 20 amino acids. So this portion up top here that is always the same, the amino group, the hydrogen, and the carboxylate group, this is what we call the backbone. And this is what I will refer to when we start talking about bringing these together to make peptides and to make proteins. The backbone will always be the same. You're always going to connect a nitrogen C-alpha carbonyl, or COO minus, and then another nitrogen, a C-alpha, the COO minus. Always, always, always. You'll see that over and over and over again. So the only difference is between amino acids. If I gave you just a backbone, you could not tell me what amino acids they are. But you would have to see the R groups to be able to determine what amino acids are present. So here's the thing. If you take a look, the amino group and the carboxylic acid group is, if you notice, you could either have it COO minus or COOH. So either a carboxylate ion or a carboxylic acid. This depends upon pH. And here's the thing. As I've drawn this on the board here, NH3 with a positive formal charge and a COO minus formal charge, that is actually what occurs at physiological pH. Okay, so we actually have what's called here a Zwitter ion. So Zwitter is German for meaning having both. Zwitter ion means has both ions, both a cation and an anion present. What you know, though, is that when we change pH, we can actually change the protonation states. So if you remember back to talking about acids and bases, remember a low pH, right, equals a high concentration of H+, meaning things will be protonated. Okay. However, when you have a high pH says we have low concentrations of H+, which means we would be deprotonated. So if you can take a look at this amino acid, if you go to lower pHs, so if this is physiological pH, a low pH would be COOH with that C-alpha and then our NH3 plus group. So if you notice, our overall charge here is +1 at low pH. At neutral pH, you have a negative and a positive, +1, -1, we are about at neutral. But if we go to a high pH, at a high pH, we're going to deprotonate. Well, we can't, there's no more hydrogens to take off here. We would not remove one from a carbon, but we will remove one from the nitrogen. So instead what we have is we have NH2 with our C-alpha and COO minus. So now here we're at -1 at a high pH. So you can see that pH can affect the overall structure of the backbone, of the amino acids. So you can look in this particular instance, right, the R group doesn't necessarily matter, but the backbone can change how many hydrogens are present on it depending on whether you're low or high pH. So again, at low pH, all of the amino acids will have their carboxylate group protonated. So your overall charge for amino acids at lower pH will be a positive charge. At high pH, right, you will have deprotonated the carboxylate group, and you will have taken off an extra proton from the NH3 group, giving you an overall minus charge. Now in the middle, at neutral pH, you have both a positive charge and a negative charge. Okay? So again, that neutral pH, you're a Zwitter ion, so you have both ions. You have both a cation and an anion present. So let's take a look. This, for example, is the structure of alanine. So in alanine, the R group is just a methyl group. So CH3. This is the amino acid alanine. Now, if you were to draw alanine at low pH, right, we would throw on extra protons anywhere that's not just a carbon. So we're not going to throw extra protons here or on the C-alpha, but we can add an extra hydrogen here. So if we take a look, we can draw alanine as CH3, COOH, NH3+, and CH. We can also draw this at high pH, right? And it would be the same as the previous example. We would just substitute in that R group, NH2, COO minus. So if you notice, the only things that change for some of these amino acids are whether you have the COO minus or COOH or NH3+ or NH2. So you should be able to, and you can get extra practice with this in the homework and problems in the back of your book, you should be able to draw any amino acid at a low pH versus physiological pH versus a high pH. So here's the thing about these amino acids. They are classified by what their R group is. So again, this goes back to those things you talked about in the general chemistry chapters, talking about whether things are polar or nonpolar. So we have, for example, here, as you can see on the slides, without drawing every single amino acid on the board here, you can see that some of these R groups, such as alanine, is just carbons and hydrogens. Remember, carbons and hydrogens are always nonpolar. So if you take a look at all these groups that you can see in these particular R group side chains, these are all classified as nonpolar because you just have hydrogens or carbons with hydrogens, carbons with hydrogens, carbons with hydrogens, right? Or, for example, here we have something called tryptophan, but you've got a big ring, which is just carbons and hydrogens, and you have one nitrogen. Or in proline, you just have carbons and hydrogen. Same thing with phenylalanine, carbons and hydrogen in a ring. If you look methionine, right, the only difference is methionine has a sulfur, but it's in between carbons and hydrogen. So if you notice, when we have amino acids that are nonpolar, okay, so nonpolar amino acids are ones that have R groups that are mostly carbons and hydrogens. So you should know your nonpolar amino acids, right, tend to have mostly carbons and hydrogens in their R groups. Which means if there are other polar groups, so polar amino acids, right, they are going to have other types of elements other than carbon and hydrogen only in their R groups. So if we take a look at these polar side chains, you can see that in these, we have a lot of oxygens or OH groups, or we have carboxylates or carbonyl groups with maybe nitrogens, okay? So if you look at those groups, you have extra pieces such as oxygen or sulfur, because if you look on your periodic table, sulfur is very similar to oxygen, or you have, again, oxygens, nitrogens. So oxygens, nitrogens, sulfurs, that's what you see in these R groups here for these polar side chains. Finally, we have different types of amino acids based on whether they can have charges or not. So these are both polar and charged, which means that you can put on a proton or take off a proton depending on what your pH is. So if you have acidic amino acids, so an acidic amino acid means that you have a group such as a carboxylic acid as the R group, which can again be deprotonated to give COO minus, similar to what we saw in the backbone here. It's now just on an R group, okay? If you have basic amino acids, so if you remember back to your organic chemistry chapters, acids in organic chemistry are carboxylic acids. Bases tend to be amine groups. So you have things here like NH3+, okay? So again, it's very similar actually to the backbone where you have NH3+ or COO minus. This is an acid group. This is a base group. If you take a look at the amino acids that you have here, right, you can see for aspartic acid and glutamic acid, these guys have COO minus, okay, or COOH if they were at low pH. For things like lysine and arginine and histidine, if you notice, they all have nitrogens with extra protons on there, NH3+, or NH2, with a plus charge because you do have a double bond there. So 4 bonds to nitrogen, looking at that formal charge. Same thing with histidine, NH2, right, plus, but then you've got 4 bonds to that nitrogen. So you can take off those protons and add protons to give you the either neutral charge that you would have here or the positive charge that you would have at those lower pHs. The other thing about amino acids, not just knowing which ones are which, so you should be able to, if given the structure of an amino acid, name it, or given the name of amino acid, know what its R group is going to be. So we can actually classify types of amino acids based on how they fit into our diet. So we have what are called essential amino acids or non-essential amino acids. So essential amino acids, they basically need to be eaten. So you can remember essential, eaten, E and E, okay? So non-essential amino acids means that we basically can make them from pre-existing other amino acids or other pieces if needed. Okay, so we have certain enzymes, which we'll talk about later in this chapter, that can convert, for example, one amino acid to a different amino acid so that we don't necessarily need to have that in our diet as a result. So that's the difference. Essential amino acids need to be eaten. Okay, non-essential amino acids, we don't necessarily need to eat them. Okay, so next we're going to talk about how we create peptides from amino acids.